Hello everyone and welcome back to AP Bio with AP Playground. Today we're covering topic 2.1, cell structure and function. Lots of objectives today, but we're just going to be going over the major components of a cell and how the structure and function of cellular components and organelles contribute to the overall function of each cell. And we'll also be taking a look at the endomembrane system that connects these various organelles in one function. And we have two suggested skills: concept explanation 1A, describe biological concepts and processes, and argumentation 6A, make a scientific claim. So before we get into the subcellular components, we have to discuss what a cell actually is. And a cell is going to be the building block of life. And this means it's the smallest level that life can occur at. So we know that atoms are the building blocks of everything in existence, but cells are going to be the smallest possible living organisms. And all cells have a membrane. They're going to have DNA, which is going to be the genetic material, an interior fluid, and this can be the cytosol, which is just the fluid, or the cytoplasm, which is the fluid plus all of the subcellular components within it, and then ribosomes, which are seen in both prokaryotic and eukaryotic cells. So let's take a look at the difference between the two. Prokaryotic cells they're very simple cells. They don't have many subcellular components, and they're single-celled, and they do not have any organelles. And organelles are going to be membrane-bound subcellular components. So there aren't, besides the cell membrane that envelops the prokaryote, there is not going to be any other membrane within the cell. Now, eukaryotic cells can also be unicellular, but they are more complex. They make up usually multicellular organisms. They have organelles which are membrane bound, and they have a nucleus that stores the DNA, whereas in prokaryotic cells the DNA is free flowing, as you can see here, whereas in the eukaryotic cell it's all enclosed within the nucleus. And it's important to remember that because we have these components, such as the membrane and the DNA, that all cells share, both prokaryotic and eukaryotic, that shows evidence of a common ancestor. But now let's start looking more at the different components, the subcellular components. So we're looking first at the nucleus and ribosomes. So the nucleus, which we talked about, is the storer of DNA within the eukaryotic cell, is going to have a double membrane known as the nuclear envelope that surrounds it. It's going to have DNA in the form of chromatin inside, so it's not condensed into what you commonly would think of as DNA with like the two strands. This is going to be not so firmly defined, and the nucleus produces ribosomes within the nucleolus, which is this central portion of the nucleus here. So what is a ribosome? Ribosome is a subcellular component that makes proteins. Now, it is not an organelle because it is not membrane bound, but it consists of large and small subunit made up of rRNA and protein, and they can be either free or bound. So they can either be free within the cell or bound to the endoplasmic reticulum, which we'll talk about in a second. So, of course, the nucleus is only within eukaryotic cells, but ribosomes are both within prokaryotic and eukaryotic cells. And generally, free ribosomes make proteins that are used within the cell, while bound ribosomes make proteins that are used for export. So they're sent out of the cell to another place in the body. And this can be done, ribosomes can move out from where they're created in the nucleolus out into the cell through nuclear pores in the envelope, which allow transport through the nuclear envelope, which is the membrane surrounding the nucleus. So we started talking about the endoplasmic reticulum as the storage, or as a place where ribosomes can be bound. But let's now go further in depth. So the endoplasmic reticulum is a network of membranes and sacs located next to the nucleus. So it's going to be right next to the nucleus, as you can see in this image here. And there are two kinds. There's a smooth endoplasmic reticulum and rough endoplasmic reticulum. So the smooth endoplasmic reticulum is going to create lipids, while the rough endoplasmic reticulum has ribosomes bound to it, so then it will be creating proteins. So each of them have a purpose, but only the rough endoplasmic reticulum is going to have a ribosome. So if we're trying to develop a protein, then we're going to start in the nucleus. The DNA inside is going to give the instruction that we need to produce a protein, and next we're going to go to the endoplasmic reticulum where the ribosomes are bound, where they can actually create that protein. Now following that, we're going off to the Golgi apparatus, or the Golgi complex, where proteins are received, modified, and shipped. So we have proteins that are going to enter one side of the Golgi complex, and this is sort of like the post office. This is going to determine where is this protein going to go. It's going to modify it slightly, and then it's going to exit. So technically it would be entering from the cis face and exiting out the trans face. And so looking at the protein production chain, or the endomembrane system overall, we're starting from the nucleus that's giving this signal that we need to begin producing a protein, and then within the ribosomes that are bound to the rough endoplasmic reticulum, we're producing that protein. Then a transport vesicle, and we'll talk about vesicles more in depth later, but a transport vesicle is going to transport the protein over to the Golgi apparatus, where it's going to be modified, and eventually it can be shipped out of the cell through the plasma membrane. And we'll talk about all of these other components in future slides, as well as in future lessons, and a lot of Unit 2 will be focused on plasma membrane and transport of things in and out of it. So we also have a few other organelles that we need to take a look at. First of all, lysosomes. And lysosomes are what I like to call acid death sacs. They are membrane-bound sacs. Again, all organelles are membrane-bound, and they're full of hydrolytic enzymes that can digest molecules. So essentially, their goal is to get rid of broken components of the cell, get rid of components of the cell that are no longer being used or are old. So they're like the recycling machine of the cell. And they do this through these hydrolytic enzymes that can digest the molecule. And they have an optimal pH, and that helps the cell reuse old parts. And because the lysosome is membrane bound, we're able to maintain that environment of having that optimal pH within the lysosome, which allows these enzymes to work. And there's also vacuoles, and vacuoles are large storage containers, and they're generally storing food, but within plant cells they can also be contractile, and within animal cells. And what they do is they regulate the amount of water within the cell, so they can pump water out or expand to hold more water in the cell. Now, plant cells are going to have a large central vacuole. This is going to be one large vacuole in the center of the cell, and that's going to be storing the majority of the water within the plant cell. And again, that too can expand or contract. Now it's time to get to some of the more fun organelles that we will be discussing more in depth in Unit 3. First up is the mitochondria, known as the powerhouse of the cell. The mitochondria has a double membrane, and we'll talk about why later in Unit 10. But the inner membrane is highly folded. It's got what are called these cristae, so these membrane folds, and that's going to contribute to a higher surface area. We can fit more surface area within the same amount of space. And this is going to allow greater exchange of resources because there is more membrane space overall within the mitochondria. But we'll talk about that more in the next lesson. The inner compartment is known as the matrix, so the interior section, and it's going to contain DNA and ribosomes of its own that are not free or bound ribosomes. It's not DNA in the nucleus. It's going to have its own mitochondrial DNA and mitochondrial ribosomes. And the main purpose of the mitochondria is to break down food in order to produce energy, otherwise known as cellular respiration. So the mitochondria is going to take the food that you eat, and it's going to break the bonds within the food in order to release energy in the form of ATP. And we'll have a whole other lesson on cellular respiration. But now let's take a look at its counterpart. Mitochondria are located both in animal and plant cells. However, in plant cells, we also see chloroplasts. And again, just like a mitochondria, there's a double membrane. There's going to be an internal section known as the stroma that has its own DNA and own ribosomes, just like the mitochondrial matrix. But there's also going to be membrane-enclosed discs called thylakoids, these guys right here. And they're going to be filled with a pigment known as chlorophyll, and that's what's going to cause plants to be green. And a stack of these thylakoids is known as a granum, and the plural is grana. And these are used to convert light energy into chemical energy. So we're taking energy from the sun and turning it into a type of energy that plants and animals can use, namely chemical or food energy. And this process is known as photosynthesis. And once this energy has been turned into chemical or food energy, then the mitochondria can then break the bonds within that in order to create usable energy for the cell. So these two organelles work in tandem. But you got to remember that only plant cells have chloroplasts. Animal cells cannot create their own food from the sun. We have to eat. And we have all these organelles, but they're not just floating around within the cell. There's going to be the cytoskeleton, cell skeleton, protein fibers that provide structural support and motility for the cell. So they're going to hold organelles in place. They're going to keep the cell within its shape, and it's going to allow the cell to move, or even parts of this within the cell, subcellular components, to move along within the cell. And so these consist of microtubules, microfilaments, and intermediate filaments. Not so important. But I wanted to point out the cilium and flagella, and these are located on different kinds of different kinds of cells. The flagella is like a tail that allows for movement, and the cilia also help with movement, but they also increase surface area by having these hairs. So again, this is a part of the cytoskeleton that helps with movement, but also with exchange with the environment. But plant cells, they have another form of cytoskeleton. They have all of this support within the membrane, but they also have a cell wall that helps maintain the shape and protect the cell. And it's mostly made of cellulose, which we talked about in Unit 1. It's a carbohydrate, it's a sugar. But animal cells do not have a cell wall. They only have a membrane. And now we're going to take a look at all the differences between animal and plant cells. So at the very beginning of this video, we talked about the differences between prokaryotes and eukaryotes. We discussed that all cells are going to have a membrane, all cells are going to have genetic information, all cells are going to have ribosomes, and all cells are going to have an interior fluid. Now, eukaryotic cells, such as animal and plant cells, are going to have this DNA within a membrane-bound organelle, the nucleus, and also other organelles that are going to help with cell functions. But now let's compare plant and animal cells. So obviously the most obvious thing here is going to be our plant cell wall, whereas the animal cell only has a membrane. And with this wall come plasmodesmata, which are sort of pores that things can get in and out of the plant cell through. And of course, the plant cell is also going to have our large central vacuole. Animal cell is going to have a vacuole, but the animal cell doesn't require as much water, so the plant cell has this large central vacuole to store water. Animal cells are also not going to have any chloroplasts. We only see chloroplasts in the plant cell, which again give the green color that plants usually have. Whereas both cells are going to have mitochondria. You can see mitochondria in both the animal and the plant cell. And in animal cells, we have what are called centrosomes, located only in animal cells, and that's, again, assisting with structure and cellular division. So they're fairly similar. They're both eukaryotic cells. They're both going to have the endomembrane system of a nucleus, endoplasmic reticulum, ribosomes, Golgi apparatus, but there are a few differences that are important to know when we're looking at their function. Plants are able to create food using the sun through chloroplasts, while animals have to consume other animals for food, which then they can turn into energy from using the mitochondria. So that was it for our cellular organelle. Make sure you know that cells are the basic building blocks of life, the various cell organelles and their functions, and some differences between animal and plant cells. And I'll see you in 2.2 to talk about cell size.